Hello students, I am Dinesh Udupi. This is session 19 of Molecular Biology, Evolution and Ethology paper. In this session, we are going to study immunoglobulins, structure and the types of immunoglobulin. In the previous session, we studied humoral mediated immunity and cell mediated immunity. So two kinds of immune responses shown by the immune system when the body gets infected. In the humoral mediated immunity, what happens? B lymphocytes, the B cells, activated B cells, they turn into plasma cells, and plasma cells start secreting antibodies, specific antibodies against pathogen or specific antigen. These antibodies will in turn destroy the pathogen. That's what we studied in the previous session. The antibodies which are produced during humoral uh, immunity or immune response, they are chemically immunoglobulins. So immunoglobulins are nothing but the antibodies produced during the immune response. Immunoglobulins are also known as antibodies. They are glycoprotein molecules produced by the plasma cells. They act as a critical part of the immune response by specifically recognizing and binding to particular antigens such as bacteria or viruses and aiding in their destruction. The antibody immune response is highly complex and exceedingly specific. Production of antibodies against a specific antigen is one of the important event in the immune response by the immune system. Immune system produces almost 5 types of immunoglobulin molecules. Of these, immunoglobulin G, which is denoted as IgG, is the most abundant. 75% of immunoglobulins produced in the body are of IgG type. Therefore, we are going to study the structure of this immunoglobulin G in this particular session. Now, this is the diagram showing the structure of immunoglobulin G. You can see here immunoglobulin G is a Y-shaped molecule. It is made up of 4 chains, polypeptide chains. 2 chains are called heavy chains because they are big in size, and 2 chains are called light chains. You can see here this is one heavy chain and this is another heavy chain, and these two are the light chains. So these 4 chains, these 4 polypeptides are held together by means of disulfide bonds. You can see here there is one disulfide bridge or disulfide bond which binds this heavy chain and this light chain. And here also you find the disulfide bond binding this light chain and this heavy chain. These 2 heavy chains are again bound with each other. They are joined with each other by means of a pair of disulfide bonds here. Like this, all the 4 chains form a common structure. They form a simple structure, and that is antibody or immunoglobulin G. The immunoglobulin G molecule shows different regions. What we studied is it is made up of 2 heavy chains and 2 light chains. In this heavy chain and light chain, we identified different regions. You can see here this part of the antibody is called antigen binding. This is the part where the specific antigen is going to bind. And this small or light chain shows 2 regions, one variable region and one constant region. In the same way, heavy chain also shows variable and constant regions. This is the diagram, the same diagram I have shown. You can see here very clearly 4 chains, 2 light chains and 2 heavy chains. They are held together by means of disulfide bonds, and the immunoglobulin or the antibody showing antigen binding site, variable region and constant regions. In this diagram, the different regions of the immunoglobulin molecules are shown. First one is FAB, fragment antigen binding. So this is the antigen binding region of the immunoglobulin molecule. And 2, this is the constant fragment. So this portion is constant and this is the one which binds with the antigen. 3, heavy chain. It consists of variable heavy, constant heavy 1, hinge, constant heavy 2 and constant heavy 3. This is the heavy chain showing variable heavy. This is called VH because this differs from antibody to antibody. And the next part is constant heavy 1. Then there is a hinge connecting the constant heavy 1 and constant heavy 2 region. This is constant heavy 2 and this is constant heavy 3. So we have variable heavy and constant heavy regions. Constant heavy region is again divided into CH1, CH2 and CH3, and between the CH1 and CH2 you find the hinge, and because of the presence of this hinge it attains the Y shape. The same thing is here. In the light chain, this is the variable region. You can call it as VL, variable light. So this thing and this part are VL, variable light, and this part is variable heavy. This is constant light and this is constant heavy. And then we find antigen binding site, hinge regions, right? So like this, we find the different regions in the antibody. One thing we have to remember regarding the immunoglobulins, in the previous session, we understood one thing, that is, when a pathogen enters into the body is going to produce specific antibodies. That means the immunoglobulin molecule which is produced during an infection is specific against that particular antigen or pathogen. After some time, another pathogen may infect and body is going to produce a specific antibody against that second pathogen. That means the specificity lies in this particular portion. The antibodies produced against all infections, say the person is infected with 100 different kinds of pathogens. That means the immune system is going to produce 100 different types of antibodies. If you compare those 100 different antibodies, the constant region is same in all the 100 antibodies. That's why this region, constant light, constant heavy 1, 2, 3. So this region is constant in all the antibodies produced in the immune system of a particular person. Then where is the difference? The difference lies in the variable region. This is the region because this is the region which is going to bind with the antibody. Antigen. Therefore, the variation lies here. So this variation is specific against the particular antigen. Therefore, it is called variable region. So 100 antibodies show variation of amino acid sequence in this particular region. That is about the structure of immunoglobulin G. Types of immunoglobulins. As I already mentioned, as a part of immune response, body is going to produce 5 classes of immunoglobulins. Immunoglobulin G is the most predominant with 75% of total immunoglobulins produced belonging to IgG class. Other classes include immunoglobulin M, immunoglobulin A, immunoglobulin D and immunoglobulin E, IgM, IgA, IgD and IgE. These are distinguished by the type of heavy chain found in the molecule. IgG molecules have heavy chains known as gamma chains. IgMs have mu chains. IgAs have alpha chains. IgEs have epsilon chains. And IgDs have delta chains. That is the difference between the types of immunoglobulins. So remember, 5 different types of immunoglobulins are produced: IgG, IgM, IgA, IgD, and IgE. The difference lies in the heavy chain. Out of these 5, immunoglobulin G is the most predominant and it is produced in highest percentage in the body whenever the infection occurs. Here, this picture or this table shows different kinds of immunoglobulins. IgA, it is found in mucus, saliva, tears, and breast milk. It protects against pathogens. And here you can see the structure also. This picture shows the structure of immunoglobulin A. IgD, it is part of B cell receptor, activates basophils and mast cells. Maybe you are familiar with mast cells, which is associated with hypersensitivity. Then IgE, it protects against parasitic worms responsible for allergic reactions. So IgE is also associated with allergic reactions. IgG is secreted by plasma cells in the blood and it is able to cross the placenta into the fetus. If you remember what we studied in the beginning of the immunology topic, there is something called passive acquired immunity. The immunity acquired by the person passively from other person. There are two methods. One is from the mother, the embryo or baby getting the ready-made antibodies from the mother, and the other one is the person getting the antibodies from a recovering patient. So in the first instance, what happens? Mother is going to transfer the antibodies produced by her immune system to the embryo which is developing in her womb. This is the first case. After the birth of the baby, she is going to feed the baby with her breast milk, and in the breast milk also, the breast milk which is produced immediately after the birth of the baby, it is rich in immunoglobulins. So the mother is protecting the baby against the infection by giving the antibodies produced in her body. And most of those antibodies transferred to the baby are of IgG types because it can cross the placenta and it can reach the embryo. The last one is IgM, immunoglobulin M, may be attached to the surface of B cell or secreted into the blood. It is responsible for early stages of immunity. That's about the structure of immunoglobulin G and the types of immunoglobulins. I hope you understood the structure and types of immunoglobulins. Thank you.